This is a production of Cornell University.
Okay, thank you very much, Mike, for that introduction. And thank you to everyone who has joined today to hear me talk about my PhD research. So I'll begin by talking about the pathogen that has been the focus of my dissertation research. It's called Phytophthora capsicum, and it's an oomycete, or a water mold, which means that when you culture it, it looks like a fungus in that it has filamentous hyphae, but it's much more closely related to certain protists like brown algae. And I'll just point out a couple of important things about its life cycle. It produces different kinds of spores. On the bottom left here, you see the asexual spores, this lemon-shaped sporangium that under water-saturated conditions releases 20 to 40 motile zoospores that are biflagellate and can actually swim and identify a new host to infect. So these asexual spores are really important for in-season spread of disease. Phytophthora capsicum is heterophthalic, which means that it has two mating types that are necessary for sexual reproduction to occur. Pretty much wherever we find this pathogen in the US, we do find both of those mating types. And that's important to know because sexual reproduction results in these thick-walled oospores that can overwinter in New York and cold climates and can actually remain dormant in the soil for, as far as we know, up to a decade, if not longer. So Phytophthora blight is really a collection of different disease syndromes depending on what plant part is infected. Most importantly are Phytophthora root and crown rot, some symptoms of which I'm showing on the left, and Phytophthora fruit rot, which you can see on the right of the slide. And as you can see, Phytophthora blight has a really wide host range that includes a lot of very economically important vegetable crops, including many solanaceous species, in particular pepper, which gave Phytophthora capsicum its name for capsicum, as well as all the cultivated cucurbits and some legumes as well, such as snap beans and lima beans. One way we know that this pathogen spreads around to different locations in New York and really everywhere that we find this pathogen is in floodwaters. Many farmers have reported seeing symptoms of this disease for the first time after a really heavy rainfall event led to their fields being flooded from adjacent creeks and streams. So with the likelihood of more erratic and extreme weather events in the future, it really seems like this pathogen will continue spreading to different farms in New York State and continue to pose a big challenge for vegetable growers. Phytophthora blight is kind of notoriously difficult to control. As a water mold, it really likes wet conditions. So one thing that's really important and recommended to farmers is making sure to implement any cultural practices that promote good drainage to deny a good environment for the pathogen. Fungicides have been used historically, but there are issues with resistance to some of these chemicals. And later in my talk, I'll talk a little bit about one of those in particular. And really resistant cultivars are in high demand, and that is what motivated my PhD research. So there are different challenges in resistance breeding in different host crops. In the second part of my presentation today, I'm going to be talking about resistance breeding in pepper. And in pepper, there are several different landraces that have been characterized for having really strong resistance to Phytophthora blight. And they've been used in breeding varieties that are available commercially that have pretty strong resistance. But a big issue that we see is that these resistant cultivars perform differently when inoculated with different isolates or strains of Phytophthora capsicum. So a major challenge in pepper breeding is breeding for durable resistance. In squash, compared to pepper, breeding is a little bit more in its infancy. There's much less known about resistance in squash. And unlike pepper, there haven't been any sources of complete resistance described in any species of cucurbita. For the most part, commercial cultivars are highly susceptible. However, we know from work that people have done screening germplasm collections and also evaluating commercial varieties in field trials that there is quantitative variation for root and crown rot resistance that breeders can take advantage of. So that's what I'm going to talk about in this first part of my presentation. A project began to breed for increased root and crown rot resistance in Michael Mazurik's squash breeding program in 2011, several years before I started as a graduate student. It was a collaboration between Michael's lab and Chris's lab. And when I first started as a graduate student, I was really happy to be able to participate in this. And the strategy that was employed was fairly straightforward, and I'll talk about that a little bit. So Michael and some other members of his lab identified a couple of accessions of squash that seemed promising as sources of partial resistance to Phytophthora root and crown rot, including this squash you see on the left of the screen, which is a Middle Eastern type squash, sometimes called a gray zucchini or a vegetable marrow. So these accessions were crossed with more horticulturally adapted lines representing different market classes. And then surrogating populations were inoculated as seedlings in the greenhouse, which you can see here with the spore suspension of Phytophthora capsucea. And it would really only take a week or so for a tray of squash seedlings that looked like this to begin to look like they appear in this image at the bottom here. So getting high disease severity is not an issue with this pathosystem. And maybe a week after inoculation, the most resistant individuals, the ones that were still alive, would be rescued with fungicide, self-pollinated, and this process would be repeated for several generations to develop inbred lines. So I collaborated with Kyle LaPlante in Michael's lab to evaluate some of these breeding lines in field trials in 2017 and 2018. And what we found was that the phenotypic selection for Phytophthora root and crown rot resistance really works. If you look on the left and look at the top, you'll see a commercial zucchini check. These photos were 2 weeks after inoculation. If you can't see anything there, it's because all the plants are dead at that point. And if you compare that to PI 615089, that's that source of resistance I mentioned earlier, or one of the breeding lines that was generated as a result of this work, you see really strong differences. And when we compare the mortality of some of the entries in these trials 30 days after inoculation, you'll see that 3 of the breeding lines that we included in these trials were among the most resistant entries evaluated. Those are represented in green here. And compared to their partially resistant parent, this PI 615089, they performed comparably or even better in some cases. You'll still see, though, that in 2018, the incidence of mortality a month after inoculation still reached over 50% in a couple of these breeding lines, showing that there's still room for improvement. One of the things that we noticed with these lines is that they showed, superior resistance to root and crown rot in the early stages of resistance following inoculation, but they still often succumbed to lesions on the vines as mature plants, or this necrosis of the shoot tips that Phytophthora blight can also cause. These lines also lacked some of the horticultural qualities that would make them suitable for production in terms of either summer squash or winter squash varieties. So it became clear that one of the issues with the greenhouse seedling inoculation strategy, and that it's somewhat inevitable just due to the high level of lethality that this pathogen is able to cause, is that when you inoculate a population of 200 or 300 individuals and end up killing 95% of them, there's not much genetic variation remaining in the population to select for other traits. So it became clear that markers could really help enrich for resistance genotypically, and then leave some variation to be selected upon phenotypically in breeding in the future. So this is the strategy that we employed to identify markers. We crossed Dunja, which is the susceptible zucchini I showed earlier that really has really high quality as a zucchini variety, with one of those breeding lines, New York 21, developed a large F2 population, and then basically just filled a greenhouse with F2 seedlings. We sprayed those seedlings with a suspension of Phytophthora capsicum zoospores, and then I walked through the greenhouse 3 days later, identified the plants that were first starting to wilt, took leaf punches from those plants, targeting around 15% of the population, came back another 5 to 7 days later, identified the plants that were still alive at that point, took leaf punches from those, and then we whole genome resequenced DNA pools from those selected tissue bulks to around 70 to 100x coverage to try to find regions of allele frequency differentiation between those bulks in a bulk segregant analysis approach. And this was repeated twice for a total of 2 independent selections of susceptible and resistant plants. So here I'm showing how we are able to use that data to identify QTL. So in these plots, each point represents the allele frequency of the allele inherited from the resistant parent at a SNP with the location of that SNP along a particular chromosome reflected in the x-axis. And SNPs in the susceptible pool are colored orange, and in the resistant pool are colored blue. And the lines are window smoothed averages. So if you look, for example, at chromosome 5, you'll see that in the middle and the end of the chromosome, the allele frequencies in these 2 pools don't deviate from one another. But at the beginning of the chromosome, the resistant pool is enriched for the allele that was inherited from the resistant parent, showing some convincing signal of some polymorphism in that region affecting the trait that we were interested in. We also saw examples of the opposite, where the resistant pool was enriched for alleles coming from the susceptible parent, showing that in this population, both of the parents were sources of alleles that conferred increased resistance. So there have been many different statistical methods that have been proposed in terms of performing hypothesis testing and localizing QTL using this really messy sequencing data from a bulk segregant analysis approach. We used one called multipool. And what we first did was we compared as a null comparison the susceptible pools, for the susceptible pool in rep 1 to the susceptible pool in rep 2. And we did the same thing with the resistant pool in rep 1 and the resistant pool in rep 2. And when we do that, we saw a maximum LOD score of around 2.6 from that null comparison. So we used a pretty conservative threshold of a LOD score of 4 for detecting QTL in the susceptible versus resistant pools. And when we do that, we find 5 regions across the genome where a LOD score of 4 was surpassed in both reps 1 and rep 2. And those were at the end of chromosome 4, beginning of chromosome 5, the middle of chromosome 8, and on chromosomes 16 and 12. I'll just point out that the QTL in chromosomes 4, 5, and 8 featured resistant alleles inherited from the resistant parent, and the chromosomes 12 and 16 QTL featured resistant alleles that came from the susceptible parent. So this approach worked pretty well in terms of the fact that we identified a really consistent signal at 5 regions between 2 independent experiments. And it was also appealing in the sense that it was pretty straightforward. We just inoculated a large population of seedlings, found the most extreme individuals and sequenced them. But a drawback with the approach is that we couldn't estimate effect sizes of the QTL. So in order to do that, and also to validate markers in those regions, we developed an F2-3 mapping population. So the F2 parents of these F2-3 families came from 2 different cohorts of individuals. 158 of them were from remnant seed of this population, and I'm calling those random families. And then 18 of those F2 parents were the F2 individuals that were the most extreme survivors of that 14,000 plant bulk segregant analysis screen. So these were the only seedlings that were still alive around a month after inoculation and that survived to produce viable seed after being self-pollinated. So when we look at the distribution of the disease resistance levels of these F2-3 families as reflected by their relative area under the disease progress curve, which is on a scale of 0 to 100, 100 being most susceptible and 0 being most resistant, we see almost a normal distribution. We see some transgressive segregation where some families were either more resistant or more susceptible than their parents. And we see that the F1 of the cross was approximately intermediate between the parents. When we divide these F2-3 families based on the cohort that their F2 parents came from, we see that the selected cohort of F2 individuals resulted in a shift of the median of their F2-3 progeny towards being more resistant, but we still see quite a few F2-3 families that were derived from these selected F2 individuals that were really highly susceptible, which kind of showcases one of the issues in these seedling screens and evaluating progeny on an individual plant basis is the error associated with phenotyping and the possibility of advancing individuals that really genetically are not superior for resistance. So when we look at markers that are tagging these QTL, we see that their effects are entirely consistent with what we identified using bulk segregant analysis. So the QTL in chromosomes 4, 5, and 8, where the resistant allele came from the resistant parent, you can see that having a copy or 2 copies of the resistant parent-derived allele results in lower disease severity in the F2-3 families on average. We can also see different types of gene action. It seems like the chromosome 4 QTL behaves additively. At chromosome 5, the allele-associated resistance appears to be dominant, and at chromosome 8, it seems like resistance is recessive. And then on chromosomes 12 and 16, we see the opposite pattern. These were the QTL where the allele associated with resistance came from Dunja, and we see that the effects on the F2-3 families is totally consistent with what we saw in the bulk segregant analysis. I'll also point out that the proportion of variance explained by these markers is relatively low, not exceeding 10% in any case. However, collectively, it seems like markers in these regions can predict F2-3 phenotypes fairly well. So you'll see that there's a pretty strong relationship between the number of QTL that are homozygous for the allele associated with resistance in the F2 individuals and the disease severity of their F2-3 progeny. We also tested the predictive ability of some different models using a cross-validation approach. And when we do multiple linear regression, just using one SNP in each QTL marker, we get cross-validation prediction accuracies that are almost as high as whole genome prediction models using 600 genome-wide SNPs. So it seems like these 5 QTL alone can do a pretty good job in terms of predicting disease resistance levels. We also use the F2-3 phenotype data and the genotype data of the F2 parents to map QTL de novo. And when we do that, we again find the same 3 QTL that we identified via bulk segregant analysis on chromosomes 4, 5, and 8. We don't detect the same QTL where the allele associated with resistance came from Dunja, and we detect a different QTL, in this case on chromosome 19. And I thought it was pretty remarkable that in terms of the locations, the most likely positions of QTL in chromosomes 5 and 8, the 2 methods, bulk segregant analysis and multiple QTL mapping, only differed by 10 kilobases in the case of chromosome 5 and 50 kilobases in the case of chromosome 8, showing a really high consistency in terms of the resolution or localization of these QTL. So these markers still need to be tested in different populations and in different environments, but they really seem promising for marker-assisted selection in this population or in similar germplasm sharing a resistant parent. And the way that I see this happening is an enrichment approach where seedlings would be evaluated genotypically for these QTL markers, either possessing the resistance allele in a heterozygous state or homozygous, and then they could be evaluated, or the progeny could be evaluated in the field for additional traits of interest to select simultaneously for Phytophthora root and crown rot resistance, as well as some other important traits in squash. So I'm going to move on and start talking about pepper. As I mentioned earlier, in pepper breeding, one of the major challenges is durability. And you can see this felt relatively easily just with this example I have on the left. So this is a resistant hybrid, bell pepper hybrid called Paladin. And I inoculated it with 2 isolates from our culture collection. And you can see that the resistance of this variety really depends on what isolate it's being inoculated with. A lot of work has been done in pepper in terms of characterizing the genetic basis of resistance to Phytophthora blight. QTL of both major and minor effect have been detected, but there's really been no research on genetic variants in the pathogen that are associated with virulence. We also don't really know the molecular or the genetic basis of interactions. You know, are there classic gene for gene interactions between R genes and avirulence genes? None of that has been characterized. So the way that I became interested in this project was several years ago, we were just screening isolates from our culture collection in order to find ones that were more aggressive or more virulent to use in pepper field trials. And I found it pretty fascinating. One, just the morphological variation among these isolates that you can see on the left here. And 2, when we inoculate them onto just 3 representative bell pepper cultivars, we see almost a quantitative distribution in terms of the levels of disease they can cause on those bell peppers. Also, of these 25 or so isolates, you can see that 6 of them caused at least some level of disease on that resistant bell pepper hybrid Paladin. So it seemed to me like if these isolates share an allele at an avirulence locus that allows them to escape detection by some resistance gene in Paladin, we should be able to map that locus using association mapping. So to do that, first we had to expand our collection of isolates. And I'm going to take a little detour from the story about the peppers to talk about our characterization of this collection of isolates that we assembled. So in total, we collected around 250 isolates that came from 24 sites in New York State, and then a handful that were sent to us by collaborators in other states. We characterized them for their mating type and their sensitivity to the fungicide mefenoxam, which has been used pretty effectively since the 1970s, but more recently, insensitivity to that chemical has been arising around New York State and really around the world. So we genotyped these isolates with genotyping by sequencing, identifying over 100,000 SNPs. That's more than 1 every kilobase on average. And that genetic marker data revealed that these 250 isolates actually represented around 130 unique genotypes. And that's because of the mixed reproduction, sexual and asexual reproduction. It turned out that about half these isolates were clones of other isolates from the same field site. When we look at the population structure of these isolates, either in the principal components analysis on the left or the neighbor joining tree on the right, we see pretty strong genetic differentiation between isolates that came from different field sites. So in these plots, the region of New York is assigned a different color, and then within each region, isolates from different fields are given different symbols. And you'll see that isolates from the same field tend to cluster together. Even if you look at these purple triangles and circles in the neighbor joining tree, which represent two farms in Erie County in Western New York that were a couple of miles away from each other, you'll see pretty strong differentiation between the pathogen populations at these two farms. This is consistent with what we know about how Phytophthora capsicum moves around. It's not dispersed in the air, but requires long distance transport via pretty rare flooding events or by farmers inadvertently moving it between locations. So we use this collection of isolates as a genetic resource to map a couple of traits. Mating type, as we expected to find, we had isolates of both the A1 and A2 mating type, pretty much at all the field sites where we collected samples. We had a pretty equal distribution of A1 and A2 mating types, so it seemed like it would be pretty tractable to identify via GWAS, the genetic control behind mating type. And we were unsurprised to see a large signal on scaffold 4 of the Phytophthora capsicum reference genome. And this has been mapped before using genetic crosses, and the same genomic region has been implicated, so we were happy to confirm those results. And we were also happy to confirm previous hypotheses that suggest that mating type in Phytophthora capsicum, and perhaps in oomycetes in general, is inherited analogously to an XY sex determination system with a heterogametic and a homogametic sex. We found that all the A1 isolates were homozygous at the significant SNPs in this region, and all the A2 isolates were heterozygous. So mefenoxam sensitivity was less evenly distributed among the different sites. You can see, for example, this location in Western New York featured almost entirely intermediately sensitive or resistant isolates, whereas fields in Ontario County included mostly sensitive isolates. Still, we were able to find a region of the genome associated with sensitivity to mefenoxam. And this is the first, these are the first results of any experiment mapping mefenoxam sensitivity in Phytophthora capsicum. So mefenoxam is known to interfere with the production of ribosomal RNA in oomycetes, which led people to presume for many years that the target site is likely to be RNA polymerase 1, which is involved in the transcription of ribosomal RNA. But what we found at this locus was not any of the subunits of RNA polymerase 1, but instead a protein that's involved in cleaving pre-rRNA molecules into the mature molecules that form the ribosome. So we have a hypothesis that perhaps mefenoxam doesn't interfere with transcription of ribosomal RNA, but instead downstream processing. And it seems like a pretty interesting candidate for functional validation. So we were really pleased with the results of the GWAS for mating type and mefenoxam sensitivity. And I thought the same approach would probably work for virulence on different peppers. So we went ahead and identified 117 isolates of those clone-corrected isolate set that sporulated well in culture, and we were able to use them to make inoculum. And then we inoculated those onto 16 pepper accessions that I'll talk about in a little bit more detail in a split plot design, where each tray was inoculated with an isolate, and then the trays were randomly divided into plots of different pepper accessions. And this was done in batches or incomplete blocks that had 3 check isolates to measure block-to-block variation, with a total of 2 complete replications. So these are the peppers that we decided to include in this experiment. We included 2 different sources of resistance, CM334, which is a landrace of serrano pepper from the Mexican state of Morelos, that's been characterized really extensively for its very strong levels of resistance to Phytophthora capsicum. And another hot pepper called perennial, which is an Indian landrace that's also been used as the parent in some mapping populations. We included bell pepper hybrids that were developed for the eastern United States, where the isolates of the pathogen that we had in our collection were from. And these included really highly susceptible red knight, Aristotle, which is not listed as resistant in seed catalogs, but we know from field trials shows a low level of disease resistance. And then 5 bell peppers that are marketed as being intermediately resistant or tolerant. We also included 6 rills from a cross between CM334 and a susceptible jalapeno variety that have been used by others for race typing Phytophthora capsucea isolates. So what we found after about a year of phenotyping was a little surprising in some ways and unexpected in others. So red knight, the susceptible bell pepper, we were not surprised to see that most of the isolates in our culture collection were able to cause disease on red knight. It was surprising, though, that perennial, which has been described as a source of resistance to Phytophthora capsucea, really was susceptible to around half of the isolates in this collection. Early jalapeno is supposed to be susceptible, as far as I know from the literature, or have a low level of resistance, but it was more comparable in terms of the number of isolates that could cause disease on it to some of the resistant bell peppers like paladin and Archimedes. And then those rills, the differential rills that I mentioned, were resistant to pretty much all the isolates in our collection. So going forward, I'm going to show the results of some analyses only using the 8 peppers where more than 20% of the isolates could cause disease, and only using 105 isolates plus 3 checks that were able to cause disease on at least one of these pepper accessions. We also saw from doing analysis of variance a significant effect of the isolate times pepper interaction, which we also were expecting to find. So when you look at the distributions of the virulence levels of these isolates on those 8 pepper accessions, you see very different distributions. Red knight has this bimodal distribution where most of the isolates could cause high levels of disease, and then a handful caused low or intermediate levels. Paladin and Archimedes and early jalapeno showed more what we expect for resistant varieties, in that most of the isolates were avirulent or caused low levels of disease, and then just a handful were able to cause high levels of disease. And then the rest of the peppers in this experiment were kind of intermediate in the sense that maybe around half of the isolates were avirulent or lowly virulent, and then the other half ranged in terms of the levels of disease that they could cause. So here I'm showing the virulence profiles of these isolates on those 8 pepper accessions. And this slide is a little bit busy, but I will try to point out the things that I think are most important. So in each plot, I'm showing, there's a different plot for each isolate, I should say. And on the x-axis are the peppers in order from most resistant on average to least resistant on average. And then the red dots are showing the level of disease that a particular isolate could cause on a particular pepper accession. The black dashed line is showing the average for each of those peppers across isolates. So one thing that I want to point out is that for a given isolate, it's much more likely that that isolate is able to cause a higher level of disease on a pepper that's more susceptible on average. And I also want to point out that the isolates that could cause high levels of disease on the more resistant peppers like paladin, for example, if you look here at this isolate from Long Island, STK5A, all the isolates that caused high levels of disease on paladin also caused really high levels of disease on the rest of the peppers, which is kind of inconsistent with a single gene for gene model, where an avirulence gene in these isolates would allow them to escape detection by paladin, but wouldn't necessarily confer increased virulence on the rest of the peppers. Finally, I just want to point out that these isolates, which are colored by the field site where they came from, really there was quite a bit of variation in terms of virulence profiles from individual field sites. And you can see a really extreme example of that on the bottom right here with this field from Suffolk County in Long Island, where we only sampled 2 isolates, one of which caused high disease on all the pepper accessions, and the other one was pretty much avirulent or lowly virulent across the board. So we tested the association between population structure and virulence profiles more explicitly. Here I'm just showing the principal components analysis again of the genetic marker data. When we broke those into 5 genetic clusters using k-means clustering. And we found when testing for differences in virulence between clusters that there was no difference between isolates in terms of virulence on any of the pepper accessions or in terms of their average virulence levels, with the exception of red knight, although there was quite a bit of variation within each genetic cluster as well. We did find, though, that multivariate virulence phenotypes showed different signals of differentiation between these genetic clusters. So on the left, I'm showing a principal components analysis of the phenotypic data, and it's colored again by the genetic cluster. So I'll kind of walk through what these different phenotypic principal components are reflecting. PC1 explains most of the variation, 67%, and that's really highly correlated with the isolate main effect, or just average virulence. And again, no differentiation between different genetic clusters. PC2 is reflecting virulence stability. So if you were to regress the virulence levels of the isolates on the average virulence levels of the peppers, like in stability analysis in G by E interactions, you would find that that slope is really correlated with this PC2. And again, no differentiation between the genetic subpopulations for virulent stability. So PCs 3 and 4 explain less of the phenotypic variance, and what they mean biologically is a little bit harder to interpret, but I think they're kind of interesting to dive into. So PC3, if you look at the loadings of the peppers on that principal component, you'll see that red knight and Aristotle have strong loadings in one direction, and then the intermediately resistant peppers, perennial, revolution, and vanguard, have high loadings in the other direction. And we see, looking at principal component 3, sorry about that. We see that this principal component really differentiates isolates from cluster 3, which are the isolates from Western New York, from isolates in cluster 2 in Ontario County. If you look at the isolates with the most extreme scores along that principal component, you can really see what I mean in terms of their resistance levels on these intermediate peppers compared to the more susceptible peppers. So I find it pretty remarkable that while there's no differentiation in terms of their virulence on individual pepper accessions, if you control for their virulence on the really susceptible peppers, then you do see differences in terms of how much disease they can cause on some of the intermediately resistant peppers like perennial, vanguard, and revolution. PC4, we see a similar story, although the differentiation is much slighter. There still is differentiation between clusters 2 and 3 and 4. And what this PC is reflecting is the virulence on that Indian hot pepper perennial compared to a couple of these intermediately resistant hybrids, Vanguard and Revolution. So we then did a genome-wide association study for virulence in this isolate collection, using as phenotypes virulence on each of the peppers individually, as well as scores along the phenotypic PCs. And we only found one SNP that was significantly associated with any of these traits. And that was this SNP on scaffold 39 of Phytophthora capsucea that only surpassed a 10% false discovery rate threshold for virulence on perennial, red knight, and revolution, but showed really consistent effects on virulence on all 8 of the peppers, explaining somewhere between 5 to 20% of the phenotypic variance. This SNP was inside a putative effector protein with the RXLR amino acid motif, which we know are proteins, secreted proteins that are known to play a role in virulence. So the location of the SNP in the genome provided some biological context that gave us higher confidence in its statistical association. But still, One of the points of this experiment was to identify variants that were associated with virulence on some pepper varieties, but not others. And we weren't able to identify any. So it's unclear why exactly that is. We know that these interactions exist. It's possible that they're of small effect and we don't have the power to map them, or maybe they're independent mutations that occur in these different field sites or subpopulations that we can't identify via GWAS. So it seems based on these data that host specialization or race differentiation is more quantitative than qualitative in magnitude. Again, we don't see patterns where individual isolates show really high virulence on individual pepper accessions, but average virulence on the remainder. Again, the isolates that cause really high disease on paladin caused high disease across the board. It's possible that in other locations, there are more, there are stronger gene for gene interactions. The differential RILs that were uninformative in our population have been informative for others, suggesting perhaps that there are avirulence genes that are polymorphic in other Phytophthora capsicum populations, but not in the ones that we characterized. And finally, that alternative approaches might be necessary for dissecting virulence genetics in Phytophthora capsicum, perhaps using biparental crosses instead of GWAS, given our inability to detect variants that might be either of low effect or rare in the population. So just to sum up the research that I presented today, in resistance to Phytophthora root and crown rot in squash, we show that there are loci of small effect that are involved. And we think that the markers that we discovered that tag these loci in some of our breeding populations really have the potential to make incremental gains for Phytophthora root and crown rot resistance while selecting for other traits of importance. In pepper, we do see that there are interactions between pepper and pathogen isolate. The relative levels of resistance among the pepper accessions in our experiment did vary depending on what isolate they were inoculated with. But again, perhaps with New York isolates, there are no large effect qualitative gene for gene interactions. It seems like with this isolate population, resistance to different isolates can be thought of as correlated traits, and selecting for resistance to one isolate is likely to confer resistance to a wide variety of isolates from the region. And perhaps breeding programs in early stages of selection can use one or two isolates perhaps, and then in more advanced material should probably trial those with a wider variety of isolates with different virulence profiles. So with that, I have quite a few people to thank and not too much time to do it. But I'd like to really thank my advisors, Mike and Chris, and all the members of their labs, as well as my committee members, Michael and Philip. The funding sources that made all this research possible, the New York State Department of Ag and Markets and the USDA National Institute of Food and Agriculture. And there are many individual people who made all this research possible. In particular, I just want to thank Kyle LaPlante in Michael's lab, who I collaborated with on all of the squash breeding. Garrett Giles, who's a technician in Chris's lab, who somehow after a year of inoculating pepper seedlings with me, managed to maintain his sanity. And then I also need to thank the Geneva Summer Scholars who have contributed so much to all this research. Andrew Allcroft, Ella Reeves, Santiago Tijardo, Jessica Helwig, Carolina Puente Silva, and Rafael Estrada. So with that, thank you all for paying attention to my seminar, and I'm happy to take any questions that you have.
Very well done, Greg. So after this, Greg will have his B exam. So the more questions you ask, the longer delay there will be to Chris and I and Michael and Philip asking more questions to Greg in his B exam. So please feel free to use the chat. Greg, if you want to open the chat, or I can read the questions to you, or you can unmute yourself and ask a question as well. Fantastic work, Greg. Congrats from Rebecca Nelson.
Okay, thank you.
This is from Larry Smart, Greg. Can you comment on the future of grafting in cucurbit production and the potential for cucurbit rootstock breeding for soil pathogens?
Sure, that's an interesting question. In particular, interspecific hybrids between Cucurbita machata and Cucurbita maxima have been used as rootstocks in watermelon production, mainly for resistance to fusarium, but people are also interested in them conferring resistance to Phytophthora capsucea. I don't know of any grafting in terms of squash production. And like I said earlier, there hasn't been much characterization of, some of these wild relatives of squash in terms of the resistance to Phytophthora root and crown rot. But I think that's an avenue that's really worth exploring. I should also mention that bottle gourd is used as a rootstock for watermelon for fusarium. But as far as I know, we don't really know much about how these rootstocks perform in terms of their Phytophthora resistance.
Okay, any follow-up from Larry? Okay, maybe not. Okay, this question here is from Sean Murphy. Such great work through all the years. The industry now has answers to help them with breeding for future resistant cultivars for PCAP. Congratulations.
Thank you, Sean. Appreciate it.
Okay, regarding the correlated selection for responses to different isolates of the pathogen, you mentioned on your last part, are the isolates genetically related? What about the effect of environment on isolate composition?
So the isolates that we used in that experiment were all genetically unique in terms of the fact that they were, not clones of each other. However, some of them are more related to each other in the sense that they came from the same field site. And it seems like their degree of relatedness is not necessarily too associated with the level of disease they can cause in individual pepper accession. But I think it'd be worth it to follow up with more isolates collected outside of New York State. We only had a small handful from the West Coast of the United States. And I think it's important to know if those pathogen populations have some avirulence alleles that are different than what we saw in this collection. I hope that answers your question.
Okay. What would happen if one used a mix of the isolates? Does one isolate typically dominate based on the susceptibility of each plant variety to each isolate? Is using a mix something that might be useful, time saving? Geez, Greg, why'd you spend all that time? No, just kidding, of course, Greg.
That's also an interesting question. And I don't really know the answer to that question because we have not used a mix of isolates, although other breeders do that. We have refrained from doing that because it makes it more difficult to replicate trials or results if you don't know, which pathogen in that mix is going to end up dominating on whatever it's being inoculated onto. But given the different virulence profiles that these isolates have, maybe that's a good idea. It could be useful in pepper breeding in terms of easily applying a mixture of, isolates with different virulence profiles.
Travis says the answer is 6. You should combine 6 and then use that.
Good to know for the future.
Oh, sorry, collecting data. Okay. Nice. Okay. How amenable are peppers, cucurbits to CRISPR type transformations?
Cucurbits, I think, are very amenable to transformation and going through tissue culture. Peppers, I know a little bit less about in that regard, but I believe they're pretty amenable as well. So I think both of those. I think in both pepper and cucurbits that CRISPR is really something that could be used successfully.
On that topic, maybe I'll ask a question, Greg. So for that one locus, that effector gene you had identified in the Phytophthora population, what would you do to confirm that's actually causal in Phytophthora?
First, it would be worth, sequencing that gene in a handful of different isolates with different virulence profiles and seeing if we could find any mutations that are, are predicted to have a functional effect on protein structure, and seeing if those variants are associated with the phenotype. And if it seems like there are promising polymorphisms that, co-segregate with virulence, I think a CRISPR approach in Phytophthora capsucea would be the best option. And it's been used in Phytophthora capsucea and other oomycetes to validate some fungicide resistance mutations, for example.
Yeah, I was going to ask how difficult it is to do an allele swap in Phytophthora.
As far as I know, it's very doable, and people have done it.
Cool, thanks. Well, I don't see any more questions. I do see a lot of great jobs, Greg, in the chat.
Thank you, everyone.
Nice job, Greg.
Thank you, Holly. I appreciate you joining in. And thank you for everything you've done over the years to make all this possible, too.
Oh, it was good. I feel like I brought you into the dark side of plant pathology.
I think that's fair to say, yeah.
All right, so with that, let's continue to thank Greg. And now we will take Greg to his B exam.
This has been a production of Cornell University, on the web at cornell.edu.